Okay, so this is more chemistry. This part's more of the biochemistry part. So let's get into it. So biochemistry is more like just the chemistry of living things and how they work. And so we can deal with inorganic stuff. That means non-carbon based, like water, salts, acid, base. And then organic does not mean like what apples you buy at the store. It means carbon based, and that's carbs, fats, proteins, nucleic acids. Um, so we're going to talk a lot about carbon. Carbon's super special. It's as special as water. So they should both be special in your heart. So here's stuff about water. Most of our cells and bodies are made of water. The percents vary, but water has really special properties because of the, mostly probably all because of the hydrogen bonding that I talked about in the last one. So water has a high heat capacity. That means that it can change. It takes a lot of energy to change its temperature, higher, hot or cold. It has high heat of vaporization. That means it takes a lot of energy to evaporate or boil it because of the hydrogen bonds, both of those. It has polar properties. That's because of the oxygen, the electronegativity of the oxygen, but also kind of because of the hydrogen bonds. It's reactive because of the polarity, and then it serves as cushioning, and that's because of the hydrogen bonds, because the water is interacting with, the molecules are interacting with each other and giving you that tension. Not tension, the, what's the opposite of tension? Whatever the opposite is. Okay, so high heat capacity, it takes a lot of energy to heat or cool, which keeps our body temperature pretty buffered. High heat of vaporization takes a lot of energy to boil or to evaporate, so that's what allows sweat to work. It's polar, it's going to dissolve other polar molecules, and it's going to dissolve ionic, because remember, ionic and polar are like sisters. Ionic is like extreme polar, so if it's ionic, it's probably going to be dissolved by water, maybe. So here's what it looks like when you dissolve water or dissolve salt. The sodium ions are going to be surrounded by the oxygen of the water, and then the chlorine ions are going to be surrounded by the H's because they're opposite charges. So that's how salt dissolves. It gets ripped apart. Even though salt's like pretty strong, right? Like it's hard and strong. When you stick it in water, that's its kryptonite. It's going to dissolve. So water also assists in reactivity and then cushioning, like we said. Okay, salts have some unique properties too. They're ions. Salts are generally ions. They're made of cations and anions, which we learned about, but not all cations and anions make up salt. So the cations are generally metals to make a salt. So H plus and OH don't count because that makes water, and the H plus is not a metal. We also call them electrolytes sometimes. Hold on. And so that's because they conduct electrical currents. And these are really important in muscle contractions, nerve impulses, like all kinds of things. And these are some salts that we need. Acids and bases are really important too. So acids have hydrogen ions. That's something you should memorize. Acids have hydrogen ions. Bases have OH ions. Acids have a pH of less than 7. And there's some really important ones that we'll talk about, like hydrochloric acid, you might have heard of. That's in your stomach. Acetic acid, you'll see it abbreviated as HAC. Carbonic acid is also really important. That's when CO2 and water connect. And then bases are proton acceptors, and they generally have an OH minus. So if you take the H plus from the acid and the OH minus from the base, you get water. Some important bases in your body are bicarbonate ions, which are kind of like the carbonic acid, and then ammonia. And ammonia doesn't have an OH, so it's a weird one, but it does still accept H plus ions. So it follows a different rule. pH scale we use to measure pH, and if you have more H's, that's more acidic, and that's going to have a pH of less than 7. And then if you have less H's and more OH's, which are the opposite, you're going to have a pH of higher than 7. And it only goes from 0 to 14. And 7's right in the middle, and that is neutral. And every time you go up 1 on the pH scale, that's a 10 times difference. So if you go from a 1 to a 3, that's 100 times difference. So acids here they are 0 to 6.99 technically, just less than 7. Neutral is 7, and then bases are 7.01 to 14. And it only goes 0 to 14. It's a mathematical thing. You use logarithm and stuff. So here's your scale. Blood is about a 7.4. That's an important number to know. So put that in your notes. And then things that are lower, you can't see this, but milk, black coffee, wine, pop, lemon juice, those are all acidic. But remember, every time you go down 1, it's 10 times more acidic. So like the difference between a 3 and a 1 is 100 times. And then if you go up, egg whites are 8, bleach is 9.5. If you get up to like Drano, oven cleaner, those are really high. Both ends of the spectrum can burn you and hurt you. So both acids and bases. Bases are usually slippery feeling like soap. And then acids are like rough feeling. Like think about putting lemon juice on your hand or vinegar. It kind of has a rough feeling, right? And then bases have a smooth feeling, like a slidey feeling. But don't be rubbing it on your skin. If you put an acid and base together, you neutralize them because if you put the same strength and amount, they're opposites, and that's because look at this equation here. The OH comes off of that sodium hydroxide, that first molecule, that is the base, and then the H comes off from the HCl, the hydrochloric acid, and that OH and H come together and they make water. So that's why it neutralizes, and it's a pH of 7. And then with the remaining stuff, you're going to make salt. NaCl is salt. So when you do a neutralization reaction, you're making some kind of salt, depending on what acid and base you put in, and you're making water. Buffers are special compounds that resist big changes in pH. So they keep the pH kind of steady. So they're things like baking soda and vinegar. Those, like acetic acid is vinegar, and bicarbonate is baking soda, and we have those in our bodies. They keep our pH really constant. Water is really bad, like pure water is really bad at keeping pH constant. pH is going to swing way out of control. But if you put a buffer in there, it keeps it really narrow and it keeps it in homeostasis. Okay, so here's homeostasis. Yeah, you got to keep it in that range. Blood's supposed to be 7.4. It can go up and down, but if you get lower than like 6.85, that's super bad. So we have a really tight, small range of proper pH that we maintain ourselves in. Okay, so this is about synthesizing and synthesis and hydrolysis. So what makes carbon so special is a few things. One thing is it has those 4 outer electrons that you probably saw in the last section, like last PowerPoint, and that means it can form 4 bonds with other things, and then it can bond to itself and make long chains or rings, or it can bond with other things. It's super versatile. It's like Legos. And it shares electrons. It does not do ionic bonding. It only shares. And so carbon makes up carbs, lipids, proteins, and nucleic acids. So it's like the building block of all of those. When you are making those 4 macromolecules that I just mentioned, you are making polymers, except for lipids. Lipids are not technically polymers, but carbs, proteins, nucleic acids, those are polymers, and they're made of monomers, which are the little pieces. So many monomers build and make a polymer. You can think of them like beads on a necklace. And they are made during a dehydration process. That's also called synthesis. Dehydration means you lose water. So when you make a polymer, you actually lose water. And then they're broken down by hydrolysis, which is the opposite, and you gain water. Hydrolysis means ripping water, and that's because the water is stuck onto the molecules to split them, to separate them. It goes on the ends of the molecules as a little nub. So let's see. Here's dehydration. This is when you're forming a polymer. You're putting 2 things together. So it's going to take off an OH off of one monomer and an H off of another monomer. So that's going to form a water. You're going to lose the water. And then they bond together covalently. And then monomers, when they're broken down, you have to add a water. And then the OH and the H are put back. And those are reversible reactions. But that's how molecules are put together and broken back apart. So dehydration is put together. Hydrolysis is broken apart. Okay, so first we have carbs. Carbs are made of CHO, remember CHO? And that's literally in the name, carbohydrate. Carbo means carbon, hydrate means water. Carbohydrates are sugars and starches, you probably know that. So CHO, they're in this ratio of 2-to-1 hydrogen to oxygen, and then they have the carbon. And there's three main classes, three types: monosaccharides, disaccharides, and polysaccharides. Monosaccharides are single, like one single ring, and they're things like glucose and fructose. And then disaccharides have two monosaccharides, so di for two, and those are like sucrose, which is table sugar, which is made of glucose and fructose. And then polysaccharides are many sugars, and those are like your starches, and they're made of many monosaccharides. So the monomer is monosaccharides. Monosaccharides, there's lots of different ones. They have different properties. Like glucose is one, ribose, deoxyribose, you'll see in DNA and RNA, and fructose is one. They can have from 3 to 7 carbon atoms. They can be in a ring or they can be a straight chain. They can kind of like unfold from the ring. So here they are: glucose, fructose, galactose, deoxyribose, and ribose. They're usually shown in a ring. So if you see a ring like this, that's a sugar. Like, you should be able to recognize that and see that that's a sugar or monosaccharide. But they can be unfolded and be straight too. So sometimes that happens, but they're going to have the same formula. Disaccharides are two sugars put together. So like glucose and fructose put together make up sucrose, which is the sugar you might put in your coffee. And you're going to make a water because that's a dehydration reaction. Other disaccharides are maltose and lactose, which is the sugar in milk. And that's why milk tastes a little bit sweet. And some people have trouble breaking down lactose into its monomers. So here is sucrose, glucose and fructose. Maltose is two glucoses. Lactose is galactose plus a glucose. And they all are slightly different. I don't think you'll see these as straight chains. I think you'll only see disaccharides as rings. So that might help. Polysaccharides are many monosaccharides together. So two examples are starch, which is in your potato and your apple. And then glycogen is kind of like a starch, but it's in your muscles. So you store carbs, polysaccharides in your muscles, and then when we eat steak or whatever, we're eating some glycogen too. Okay, so this is what it looks like. It bonds different ways to make like starch or like cellulose. Like I think of celery, the strips of celery. That's also polysaccharide, and it's going to bond in like different configurations to make the different starches or the different polysaccharides. Okay, now we'll look at lipids. Lipids are CHO, CHO, just like carbs, but sometimes they have a P, so you can write down CHOP. Lipids are fats, waxes, things like that. They include in our bodies triglycerides, phospholipids, steroids, and I don't know how to say this one, eicosanoids. And I don't know why the H's are capitalized in phospholipids. I think that's a typo. I think they accidentally were putting like pH. So don't capitalize the H's in phospholipids. So triglycerides, you might have heard, you might have had to get your blood checked for triglycerides. Those are fats when solid and oils when liquid. That's usually the words we use. Like if it's a lipid and it's liquid, we call it an oil. And if it's a solid, we call it a fat. So like your solid butter, that would be a fat. And your cooking oil would be an oil. Triglycerides, though, are really important in our bodies. We use them for energy storage, insulation, protection. They're made of 3 fatty acids. That's what the tri part is. And then they have a glycerol, and that's a sugar alcohol. So here's what they look like. Here's the glycerol, and then they have the fatty acids. And you do a dehydration reaction. All of these are made via a dehydration reaction, and you make water. They're all the same. So there's your 3 fatty acids. And I think of it like a fork. Like it kind of looks like 3 prongs on a fork. What's this showing? This is a simplified diagram. Whenever you see a wiggly line like this in chemistry and science, they're showing a carbon molecule, and each little corner of the wiggle is a carbon. They just show it like simplified. And up here on the glycerol, they're showing the carbons, but down here they're not. Okay, and then you can watch that video. So there's different types of fats, and you might be aware of this in your diet. There are saturated fatty acids. those are ones with only single bonds, and they pack together really nicely, and so they form solid fats at room temperature. So here it is, they only have single bonds in their chain, just the chain, like don't worry about the double bond at all. Only single bonds, and they form solid fat, that's saturated. And then unsaturated have some double bonds, and so the carbon chains become kinked. They can't fit together, so they're not solid, they're liquid at room temp. And then there's trans fats and cis fats. Trans fats are usually considered bad for you because we don't have the enzymes to break them down. And then omega-3 fats are good. So here's your picture of the double bond, and it causes like kinks in the chain, and it makes it liquid at room temperature. And then phospholipids are really, really important. This is what makes up our cell membrane. They're made of triglycerides, except there's only 2 fatty acids instead of 3. And they have a head and tail. The head of it is polar, the tail is non-polar, and they form this double layer, and that's what makes up our cell membrane. So here's what they look like. You've got your glycerol, you also have a phosphate and usually a nitrogen containing group, and then you've got your 2 prongs. They usually are unsaturated, like they usually have double bonds, because if they didn't, it would, the cell membrane would be like too rigid, and it needs to be like a little bit more wiggly and pliable. So here's what they look like. You might have seen these pictures. This is what all of your cell membranes, every cell membrane on the planet is made of this. Another type of lipid is a steroid. Steroids don't look like the other lipids. The other lipids had chains of carbon. Steroids are usually rings of carbon, but they're still non-polar and they're still lipids. Usually there's 4 rings hooked together. The most basic one is cholesterol, and then your other hormones are built from that usually. They're important to make vitamin D, steroid hormones, bile salts, also used in the plasma membrane, so steroids are really important. Here is a picture showing what a steroid looks like. So this is your base cholesterol. It's made of 4 rings, and each little corner is a carbon. They are showing some of the other carbons, but you can see each corner there is a carbon. This one's a double bond, but this is your base structure of cholesterol, which is a steroid, and then you can add on different parts to make different hormones. Okay, how did I say this? Eicosanoids. Pretty sure that's right. These are also important lipids. They come from a fatty acid. They're in cell membranes. They are important in blood clotting, blood pressure, inflammation, labor, and the inflammation can be blocked by NSAIDs. So you'll see these in different things, and they're important too. Okay, the next macromolecule is proteins, and proteins make up most of your cells, like the stuff of your cells. They're your hair, your skin, your nails, most of your like stuff. The structural stuff is made of proteins. Super important. They are made of mostly chons, CHON and S, sometimes P, but I wouldn't focus on the P. So mostly just write down the CHONS, chons. That's what they're made of. They do have monomers. The monomers are called amino acids, and they bond together. They have covalent bonds between the monomers, and they make your protein. They make peptides. So here's some pictures of them. Proteins are the workers. They're the main bulk of what happens in your body and in your cells. So there are structural proteins. This is collagen. You might have seen commercials about that for like lotions if you're getting wrinkles. There are also enzymes. We've had that word before. So these are catalysts that help reactions happen. Those are proteins. They are transport proteins. They move things around your body. They're hormones. They do all the work. That's what proteins are. Super important. There's also contractile proteins. This is in your muscle. This is myosin and actin, what makes your muscles move. There are proteins in your cell membrane. Do you recognize these little phospholipids here? And these are little channels or doorways in your cell membrane that let things in and out or open doors, do really important functions. There's also defensive proteins like antibodies that protect you against pathogens. So there's, to make up your proteins, there's 20 different amino acids. And those are the monomers. And you can think of the amino acids like letters in the alphabet, and then the proteins are like words, okay? So they form together with peptide bonds. So like you're putting your letters together in words. The peptide bonds are the ones that connect them, and those are covalent bonds. They're made via dehydration reactions, so you're losing a water every time you're making a peptide bond. Those are all the same mechanism. So here's a basic amino acid, and you have a carbon in the middle. You always have this NH2. That's where your N comes from in your CHON. And then you have a carboxylic acid group over here, the COOH. And then you have this R group, and this is the group that gets switched out to make your 20 different amino acids. So there's 20 different possible R groups. They all have different properties, and they cause your protein to fold up differently depending on what property each amino acid has. I think I went back. I'm not sure what happened. Oh yeah, the R groups, the 20 different R groups. So this is what happens when you make a peptide bond. You got two amino acids, you add them together. That is a dehydration reaction. You lose the water and you make a peptide bond. And that is called a dipeptide because there's two of them. And then you just keep adding them. And then you can do hydrolysis and break them apart. So when you're eating a steak and you break it down, you're doing hydrolysis and breaking them down into individual amino acids, and then you build them back up through dehydration into like your hair and your skin and your collagen and all that. There's different levels of that we look at with proteins of like how they fold and how they interact, like kind of different dimensions. The first level is called primary, and that's where we're just looking at what order are the amino acids in. And then the secondary level is how they start to fold up, because proteins have to fold up to do a job. So there's two different main shapes that they take on when they fold up. They take on an alpha helix, which is like a telephone coil, and they can take on beta pleated sheets, which is like a zigzag kind of shape. Like think of a paper being folded like zigzags. That's the secondary level. The tertiary level is when they start to fold up more complex and like take on their shape, whatever job they have. And then the quaternary is when you put multiple ones together. So this is the primary structure, and it's just each one here is an amino acid, and it's just like what order are the amino acids in. And then secondary structure starts to make alpha helices, which is this coil, and beta sheets. And then they'll fold up into a complicated tertiary structure, the third level. And then multiple of those together make the quaternary, the fourth level. Okay, and you can watch that video to help you. There's different types of proteins that have different shapes, and all these proteins have different jobs. There's probably like 20,000 genes that code for your different proteins in your body. I don't know that they know that there's necessarily 20,000. There's probably more than 20,000 proteins actually. So there's tons. They all have different jobs. They fold up in different ways. One type is a fibrous structure, and these are water soluble. Things like keratin, elastin, collagen, contractile fibers. So those are mechanical support. And then you have globular. Those are also water soluble. All of these can change shapes in the wrong conditions, and then they'll be the wrong structure, and they won't be able to do their jobs. So that's important. So globular are things like antibodies, hormones, molecular chaperones, enzymes, like the little workers, right? The ones that aren't as structural. So proteins can unfold in the wrong conditions. That's what I was just mentioning. That's called denaturation. So specifically globular proteins are like the less stable ones. Fibrous are less likely to do this, like your hair. It's going to do it. Your hair is fibrous. It's going to do it if you like bleach it, but it takes a lot to get it to change shape. So if you put globular proteins in the wrong pH or the wrong temperature, like when you're cooking an egg, that's the egg white is a protein. It's going to change it, and it's going to change the folding. It's going to change the active site, and it's no longer going to work. So that's really important, because when our bodies aren't maintaining homeostasis, they can cause stability issues in your proteins, because they're causing situations where they're not in the right pH or temperature. Enzymes are also globular proteins, and those are the catalysts. And so if they're in the wrong conditions, they're going to unfold and not do their job. But these enzymes make your chemical reactions happen. If we didn't have enzymes, your body would have to run at a higher temperature, because remember that if you increase the temperature, it increases reaction rate. So the enzymes help the reactions happen better. So you don't need as much reactants. You don't need a higher temperature. You don't need to shake your body up and stir to make sure your chemical reactions are happening. You have enzymes for that. So this is why sometimes if people are like lactose intolerant, that's because they can't make the enzyme to break down the lactose into galactose and glucose, and so the lactose makes them sick. So the enzymes are the ones doing the work and the magic. Here's some characteristics of enzymes. So most of them are called hollow enzymes. They have two parts. They have an apoenzyme and a cofactor or coenzyme, something else that comes in and helps. Sometimes it's a vitamin. That's why you need vitamins. And enzymes are super specific. They only work with certain substrates. So you have to match the enzyme to the substrate. It's not like a one size fits all. Like it's a lock and key situation. Certain lock fits a certain key, and that's it. And a key to knowing if something is an enzyme is often if the word ends in ase, ase, that's often an enzyme. So that's kind of a little clue, a hack for you. If you see that, it's probably an enzyme. Not all enzymes end in ase, but a lot of them do. What enzymes do to increase the reaction is they lower activation energy. So you don't need as much energy to get it started, to get that reaction to go. So they like prime it, right? The way they do this is the substrate is going to bind to what we call the active site on the enzyme, and that's a spot where they're chemically compatible. The substrate, you guys don't know what the substrate is yet. The substrate is the molecule that the enzyme is going to act on, like the key, and then the enzyme is the lock. So the substrate is the key that's going to fit into the enzyme, the lock. Makes sense? And they have to fit perfectly into the active site, and that's the spot where they are compatible. They're chemically and electrically compatible. And then something's going to happen to the substrate. Either the enzyme is going to put two things together and build up two things and do synthesis, or the enzyme is going to break two things apart and do decomp. That's usually the two things. And then you'll have your product is released. Here's a picture showing activation energy. So usually it's going to take like a lot of energy just to get that reaction going. And so you'd have to heat it up or add more substrate or whatever. You can think of like rolling a rock up a hill to release it. But enzymes lower that. They lower the hill. They make it easier for the reaction to happen. And here is showing like the lock and key. So here's the substrate. This is a synthesis reaction because you're trying to put these two things together. You're trying to make a dipeptide, right? You're trying to put two amino acids together. So there's your active site. They're compatible. They fit lock and key. And then the enzyme causes this bond to be formed. And then off it goes as a dipeptide. The enzyme is not used up. The enzyme is not changed. It's going to go back, like it's changed while it's doing it, but it's going to go back to normal after the reaction happens, and it can be reused. And remember, enzymes are proteins too. So in this case, you've got a protein putting together two amino acids to make a dipeptide. Okay. Okay, then we have nucleic acids. Nucleic acids are DNA and RNA. Those are your two types. They are composed of CHOMP, C-H-O-M-P, and they are made up of nucleotides. So the monomer is nucleotides. The polymer is the nucleic acid, like DNA or RNA. DNA is the double helix. It's probably everything you've learned. It's made up of deoxyribose sugar. It also has a phosphate. And then it can have four different bases. It can have adenine and guanine or cytosine and thymine. Adenine and guanine are called purines, and cytosine and thymine are called pyrimidines. Pyrimidines have a single ring to them, to that base, and then purines have a double ring. So that's the difference between them. And then adenine always binds with thymine, and cytosine always binds with guanine. That's the bonding rules. That's what they're compatible with. There it is. A always bonds with T, G always bonds with C. That's the complementary base pairing rules. That's Shargoff's rule if you learned about that in biology. Okay, so here's DNA. You can see the A's and T's, C's and G's. A's and T's form 2 bonds, C's and G's form 3 bonds, and they are hydrogen bonds. They're really weak bonds, but remember, if you have enough hydrogen bonds, they are pretty strong altogether. And this is showing you the 2 rings to adenine and the 1 ring to thymine. So the 2 ring purines always bond with the 1 ring thymine. Okay, then you have RNA. RNA is single-stranded and DNA is double-stranded. And RNA has a ribose sugar instead of a deoxyribose, and then it has a thymine instead of a uracil. And there's lots of different kinds of RNA, not just these 3, but these are the 3 that we learn about to learn about how protein is made, that you've probably heard of. So mRNA, tRNA, and rRNA are the 3 that we talk about the most, but there's a gajillion others. So you can watch this to show you about DNA and RNA. And then we have ATP, and ATP is actually like a sister to DNA and RNA. It's technically a nucleic acid. ATP is the energy of the cell. It's the molecule that the cells all get energy from. And it cycles between ATP and ADP. ATP is adenosine triphosphate, and ADP is adenosine diphosphate. And so when it's triphosphate, it has energy, and then when it's diphosphate, ADP, it's like an uncharged phone, like it's dead, and then it recharges. So here is ATP with 3 phosphates. This is what the phosphates look like. And then this is really similar to DNA. There's an adenine, we saw that in DNA and RNA, and then a ribose, we saw that in RNA. So it's basically a nucleotide from RNA, actually, and then you add 2 more phosphates to it. So we call it ATP when there's 3, ADP when there's 2, AMP you will see sometimes, and that's when there's 1. That's important in cell signaling. Okay, so ATP, that 3rd phosphate is what kind of stores the energy in that bond. If it loses one, it becomes ADP. So here's like your cycle. It's still a dehydration or hydrolysis reaction. So that's important. And then when you lose, like when you go to ADP, you're going to have a phosphate hanging out. And those phosphates are also really important for cell signaling and all kinds of things. Phosphates are golden. So here's some places that we use ATP. ATP, I think, runs about, I think the number is like 70% of our cell processes. So it is like the money of the cell, of your body. When you eat glucose, anything with glucose, it's probably going to get converted into ATP. So here ATP is being used to bring things out of the cell or into the cell. Here it's being used to contract a muscle, and then here it's being used to do chemical work. So ATP is really the money. And that's the end.